Hello guys, so my name is Carolyn and I am going to go over how COVID-19 is detected on a PCR test, and this PCR test is usually given by physicians and other healthcare providers in a clinical setting, and this test does take a few days to process in order to confirm a definitive diagnosis of COVID-19. This is very different from the antigen test, which you usually buy at Walgreens, and it's a rapid test to see if you have the COVID-19 antigen, and that is less accurate than the PCR test. But we're going to go over the PCR test today. In this presentation, I am going to go over the basic principles or definitions of what DNA is, what RNA is, how that relates to the functionality of a virus, and how a virus is able to live within its host. And then I'm going to connect that and delve into what COVID-19 is, that specific virus, how that works, and how the PCR test is able to detect the COVID-19 virus. All right, so this first slide, I'm going to give the definitions of what DNA, RNA is, just a basic overview. So every living organism, including viruses, carries genetic information encoded in molecules called DNA, which we're familiar with, and RNA. So DNA stands for deoxyribonucleic acid, and RNA is ribonucleic acid. And both contain genetic information within an organism, whether that be in humans or animals, okay? So, and this includes viruses as well. So DNA, to differentiate a little bit, the fundamental difference is that DNA is the genetic code of an entire organism. And DNA is what makes RNA. And RNA is a single-stranded part, as you can see here, is a single-stranded nucleic acid that brings the instructions from DNA into life. So RNA is what makes the proteins that are necessary to make enzymatic reactions occur and to help carry the cellular functions and help an organism live. So given the fundamental difference between DNA and RNA, that helps a cell to develop its organelles like mitochondria and to develop into tissue and then so forth. So what is a virus? A virus is a microorganism with a nucleic acid segment and a protective protein coat. So here I have an image of different types of viruses and their coating shapes. But a virus is a cell that is not living, and this contrasts to bacteria, because bacteria are able to survive on their own as they have the cellular machinery like mitochondria to sustain themselves, to grow, to proliferate, and to live on their own, sustain themselves. Viruses, because of that little nucleic acid segment, even though it's enclosed and it's able to kind of live in a host, it can only live in a host. And because of that, it needs to affect the host machinery in order to replicate itself and live. So on this slide here is just a little background on the life cycle of a virus, and it's usually in 6 steps. This is just a simpler way of giving you an idea of how a virus is able to live and make more of itself within an organism, okay? So it's going to attach within a host cell, and this can be in any organism, an animal or a human. And once this virus attaches to the cell, it's going to penetrate past the plasma membrane and travel towards, the end goal is to travel into the nucleus and make more of itself, okay? So before that, it's going to penetrate the cell membrane, go inside the cell. This coating that we talked about, the protein coating that we talked about in the previous slide, is going to release this genetic material. The genetic material is going to kind of mask itself, if you will, so that it can confuse the host cell into making more of it and to making more proteins that support this genetic material to be made as well. So here we have the proteins and more of this viral genetic material that is going to be made. And both are going to be assembled into more of these viral cells that are integrated with the host cell so that it's not recognized by the immune system to be killed. So this is assembled and released, and these other cells are basically going to multiply and mix in with the regular cells and in order to affect the organism. So this can manifest in clinical symptoms like cough and respiratory distress in terms of COVID-19. But the concept here is that the virus is going to go inside the cell, and the protein coding is going to release the new genetic material. Genetic material goes to the nucleus, makes the nucleus make more of itself and the proteins to make the viral cell again, and that will be released to infect the rest of the body. Okay, so let's go into COVID-19 now. Coronavirus is a disease that's caused by the SARS-CoV-2 virus. Now, the SARS-CoV-2 virus it basically derived from the severe acute respiratory syndrome that was an outbreak in 2002, and that virus is the genetic material with the RNA genetic material that causes COVID-19. But COVID-19 is the disease that's manifested by this virus, okay? So the SARS-CoV-2 virus is a microscopic entity with a spiked outer layer. And the spiked outer layer here is how coronavirus got its name, because these spikes are surrounding this virus and the genetic material inside, and it represents a crown, which is what corona translates to, crown. Now, these spikes are these proteins, this protein coding that, like in the virus cycle, allows this virus to penetrate the plasma membrane in a host cell, so humans, and then to go towards the nucleus, replicate itself more, and infect other cells. So now I'm going to go into the pathophysiology of COVID-19. Pathophysiology is just me explaining how the virus is able to penetrate the body and manipulate the host cell machinery to replicate itself and manifest in the symptoms that we know now today. So the SARS-CoV-2 virus is going to go into the nasal epithelial cells, and we contract this virus either by someone else sneezing or contact with another sick individual that has the virus. And once that virus is contracted, it goes and binds to these nasal epithelial cells. And these nasal epithelial cells are just cells that are cells that are meant to protect the organs, okay? They either make secretions or mucus to help with multiple things, mostly lubrication, and they also have cilia, which are these little hairs that you can see here, and they can move some foreign pathogens along, like in the lungs, to help with breathing and to prevent any mucus buildup that is unwanted within the lungs. So that's just an example of epithelial cells and a little definition there for you. But once it binds to the nasal epithelial cells, it's going to replicate itself, and it penetrates first the upper respiratory tract. The upper respiratory tract is where you breathe from the mouth and then going towards the trachea and before the lungs, okay? So that's the upper respiratory tract. These virus cells are going to go from the nose, then go down towards where the mouth is and down to the trachea, and then it's going to affect the lower respiratory tract, which is where your lungs are. Now, once it affects the lower respiratory tract, it's going to affect these alveolar epithelial cells. So the alveoli, if you can see in the lungs here where you have these branches, okay, those are called bronchi, and they branch out to these little bulbous sacs, and these bulbous sacs are called alveoli. This is where gas exchange in the body occurs. Now, the alveoli, these bulbous sacs, are going to provide oxygen for the blood to take, and the blood is going to give the carbon dioxide for the alveoli to make as a waste product. So this gas exchange is where it occurs at the alveoli. Okay, so once these alveolar cells are targeted by the SARS-CoV-2 virus, this is where a lot of the clinical manifestations occur, such as respiratory distress, wheezing, any breathing difficulties, especially in those with comorbidities of COPD, chronic obstructive pulmonary disease, and diabetes. It has a huge effect on someone's ability to breathe. And so then once this occurs, a lot of the time the virus will replicate more, and because it's replicating more, the immune response, the immune cells will recognize that something is going on and the virus is attacking the body. And once it recognizes it, it has these immune cells, these T cells, the white blood cells, attack the virus. But in attacking the virus, when they recognize these little antigens that pop up on the cell, they attack the virus, right? And once they attack the virus, the whole cell dies, okay? And the host cell releases more antigens, or these little proteins that are characteristic of the COVID-19 virus, in order for more immune cells to recognize these antigens and initiate more immune response to kill more of the virus before it takes over the whole body. So this cellular death or apoptosis of the host cells release these antigens, but it cannot, in most cases, the virus makes more of itself, and the replication and able to replicate and manipulate the cellular machinery to have more mutations, so that it's not able to be as recognizable by the immune system. So it proliferates and is able to, a lot of the time, have a worse clinical outcome for patients, and that's why it was so deadly and created such a pandemic in 2019. So this inflammatory response is what represents in fever, and it generates what is called a cytokine storm. So all this release of immune proteins and kinases that try to eliminate the virus and attempt to kill the cells, but it is not able to, and the inflammation is what causes the fever, or the body aches, and the general feeling of malaise. And ultimately it causes a lot of alveolar damage, because the inflammation is the way for it to kind of stimulate the immune system to work, but in turn it is causing more damage to the lungs. So there are two types of COVID-19 tests that are used to diagnose the virus. One is the polymerase chain reaction test, and the other is the rapid antigen test. The polymerase chain reaction test is what we're going to go over more in detail. And this test is usually done in clinical settings, because they have to measure the genetic material, versus antigen testing, which is rapid and done at home. A lot of individuals would prefer to get the antigen test and from the local Walgreens drugstore and test themselves for a rapid diagnosis. But most individuals do not know that the antigen test does not yield as accurate of a diagnosis compared to the PCR test. And this is because of what either test measures, which is different. The PCR test measures the genetic material, which is the RNA, in this case for the COVID-19 virus, versus the antigen test, which measures the proteins of the virus. So oftentimes a lot of individuals will take the antigen test and it'll rapidly say no. But some cases, the virus hasn't developed enough yet or replicated enough proteins and can be in the asymptomatic or early stages of the virus cycle. So it can often lead to misdiagnosis and people not realizing that they have the virus until later on. So the first step of the PCR test is obviously to collect the sample. Now, the COVID-19 testing process, the first step is going to take the nasal swab, and you're going to go deep down the nose. And a lot of people don't realize that the deeper you go, it's because the virus, like we said in the virus cycle, it usually goes towards the nasal epithelial cells. So that is why we're going all the way down lower in the upper respiratory tract to make sure that we get all those epithelial cells to see if there is any genetic material or RNA of the virus there, because that is usually the first place of penetration of the virus where it replicates itself and is in the quiet stage of the virus. After collecting the sample is going to be mixed in with your DNA and potentially the viral RNA, okay? So in order to isolate the genetic material, the RNA of the COVID virus, we want to extract it. So we're going to extract that RNA so that we can just focus on that component. And this involves in the lab processing other cells and other proteins and genetic material of our own that's intermixed with the viral material. So we're separating our genetic RNA of the COVID virus from our DNA and isolating it. So now that we have isolated our RNA, which is the genetic material for our COVID-19 virus, we're going to take that extracted RNA that's isolated and use an enzyme called reverse transcriptase to convert the single-stranded RNA, this little viral genetic segment of the COVID virus, and convert that into complementary DNA or cDNA. And this cDNA is going to be the double-stranded DNA that we often see first before making the RNA segment. So this cDNA serves as the artificial copy of the virus's genetic material, and the two double-stranded DNA is going to be split. Okay, so the third step is going to be the polymerase chain reaction. So we have a primer which has the segment of DNA, and that primer has two parts. One that is going to cause fluorescence, which is a light, and the other part of the primer is the part that prevents the fluorescence. So when these two parts are attached to the primer, that means that it's not going to cause a light for us to know and detect that the viral DNA is there. Okay, so the primer is supposed to attach to the viral DNA segments, and that's going to code for COVID-19. So once the primer, which has the two parts, attaches to the viral DNA, the primer is going to want to make more of the complementary DNA strands. And once these DNA strands extend, it causes one part of the primer to come off, causing the fluorescent part to just stay on there and to make a light, indicating that the viral DNA is being duplicated. And the primer is designed to specifically attach to the COVID-19 virus within our sample. So from that last step, we have two duplicate copies of the viral DNA. And so we're going to put this mixture and repeatedly heat and cool it in this PCR machine over here. And each cycle is going to double the amount of DNA in the sample from the original sample that we got from the nasal swab. And it's going to kind of allow us to understand how much genetic material was originally in the sample. So the results of the qPCR reaction are represented in a graph known as a fluorescence curve. And the fluorescence curve is just basically measuring how much fluorescence derives as we increase the amount of cycles in that procedure. And a clear and sharp curve indicates a presence of the COVID-19 virus in our sample. And the cycle at which the fluorescence crosses a predetermined threshold known as the cycle threshold provides information about the amount of genetic material in the sample. So I'll go more into that in a moment here. So our last step is when we're quantifying our polymerase chain reaction, so hence the qPCR, and this correlates to the amount of fluorescence that we're getting after each cycle. So the more cycles that we have, the more we anticipate if the individual does have the COVID-19 virus, more of that DNA being generated. Now, after each cycle, there's going to emit that fluorescence, correct? So the CT value is going to be the threshold cycle. And this value represents how many cycles it takes to get a certain amount of fluorescence. And we'll see this in the graph that's generated as we do the quantification. So lower CT values mean a higher RNA expression, which means that more viral DNA is going to be made. Given this, we can determine that the higher CT values means there's low RNA expression, and this does correspond to the values we have here for the COVID-19 virus. So the CT value of 35 or higher does mean that a person is COVID-19 negative. Under 35 means they are positive for COVID-19. So this goes hand in hand with the relationship that we just specified. So let me just recap here on the detection of COVID-19 with the PCR test. So the PCR test, we're isolating that viral RNA from the COVID-19 virus, okay? And then we're running it through the amount of cycles. The more copies of the viral DNA that we make, the more fluorescence it's going to give. The more fluorescence it's going to give, the lower the CT value. And we know that the lower CT value means you're positive for COVID because it takes smaller amount of cycles to detect fluorescence, and the fluorescence is linked to more transcription of that DNA and copying of that. So that is how we are able to detect COVID-19, and this science has been able to ultimately reduce the severity of the pandemic through just this basic science. So thank you for listening, and I hope this was easy for you to understand.